once we are finished with the types of pollination, right? I explained you already about the self-pollination and cross-pollination. Let me little bit explain you about that, and then we will learn about the various agencies involved in transfer of pollen grains. First, I told you self-pollinating plants, they are actually having no color, no perfume, no scent, right? Then cross-pollinating flowers will have all the three or one of them, right? Then some of the plants will undergo compulsorily self-pollination. Those are normally closed flowers, right? Like Camelina, right? They are closed flowers, right? That is one of the plant, but most of the plants which are open, they are called chasmogamous flowers. They undergo compulsorily cross-pollination. Sometimes if the cross-pollination fails, they undergo self-pollination. Okay. Now we will talk about the various agencies or vectors involved in cross-pollination. Yesterday I told you, the next class, the previous class I told you what are the advantages and disadvantages of self and cross-pollination. Right. Now we will talk about the various vectors involved in cross-pollinations. Cross-pollination, what are the various vectors involved? So you all know that mainly I told you already once we discussed it is about the wind and the insect. Apart from that, there are a lot of vectors. Based on the type of vectors, we classify or we name different types of pollinations. Suppose first one, if the pollination happens with the wind, we call that as anemophily. Very important this is, you have to note it down, anemophily. What is anemophily? It is the wind pollination, means pollination through wind. Okay. Second one is through the insects, we call that as entomophily. Entomophily is pollination through insects. Insect pollination. Okay. Third one, if birds are involved in pollinating, transferring the pollen grains, then such type of pollination is called as ornithophily. Ornithophily. All these are 1 mark questions, or they may ask you as types of vectors, or based on vectors how do you classify pollination. So it is ornithophily refers to pollination through birds. So birds pollination. Fourth one, other animals including humans, goats and sheep, everything, and the cow and all, when they are moving around, they bring about pollination. That is the pollination through animals. We call it as zoophily. Zoophily is pollination through animals. Animals in helping in pollination. Then fifth one, in the plants which are living in water like Vallisneria and Pistil and all, pollination happens with the water currents. We call that as hydrophily. Hydro refers to water. Hydrophily is pollination through water. Water pollination. Sixth one, sometimes the pollination happens with the help of the snails. Snails, I hope you know that, in a marshy land and all. Snails help in pollination. Then we call that as malacophily. Malacophily is nothing but the pollination through snails. Okay. Then next one, there are so many but few only I will give you. This is there in your NCERT book, so we are talking about them. Last one, in the night blooming flowers. Some of the flowers open up in the night time. Those are transmitted, their pollen grains are transferred from anther to the stigma through the bats. We call it either bat pollination or chiratoryphily. Okay, chiratoryphily is pollination through bats. Bat pollination. So this much at least you should know. What are those? Just listing them is important. Anemophily, pollination through winds. Then entomophily, pollination through insects. Ornithophily, pollination through the birds. Hydrophily, pollination through the water currents or water. Zoophily, pollination through various other animals. all those already insect I talked about, that also is an animal, but that is more common, so we are separating entomophily from rest of anemophily, sorry, zoophily. Next is about the malacophily. It is normally found in a marshy area where the snails bring about pollination. Then the last one is chiropterophily or chiropterophily. This is nothing but the pollination with the help of bats. This pollination happens in those plants that will bloom at night blooming flowers, because at that time most of the insects and all cannot move around. Therefore, night moving animals, they are called the nocturnal animals. Nocturnal animals are the night moving animals. They are called nocturnal or nocturnal, like bats. They are night, they will be moving or they are flying only at night, and they will identify the or target or the area through the echoing of certain different type of sounds. Therefore, that is the only animal which can help in pollinating these night blooming flowers. It is called chiropterophily, right? So these names you should remember and list them. Actually, this list is important, and separately you have to write one line explanation. That's it, right? You are not going to do big explanation. 1 mark question. Just what is anemophily, or what is the name for wind pollination? What is the name for insect pollination? So you have to write their names. Okay. So I hope you understood. This is very simple. Just to by heart them and understand, remember their names. Okay. Now let us talk about the characters of these wind pollinating or insect pollinating plants. So first one I will give you the characters of anemophilous flowers. Anemophilous flowers. So what are the characteristics of anemophilous flowers? So you know, first anemophilous, this has been asked for 2 or 3 mark question in any of most of the public examination question. It is give the characteristic features of anemophilous flowers. So it's very simple. Just imagine wind has to take the pollen grains. So if the plant is producing pollen grains, if at that time when the pollen dehiscence take place, that time wind blows. Through the wind current, they will fly from one place to another place, bringing about pollination. But this is a very unguaranteed point. No surety in this pollination. Why? Because wind will blow on its own. Pollination has to happen if the wind current is one direction, plant is in another direction, other plants are in other direction, then the pollination may fail. So why I am telling all this? Just because wind pollinating flowers will have to adopt themselves to release their pollen grains in air. Right? So what they should do? First thing, because there is no guarantee in pollination, they should produce the pollen grains in the large numbers, enormous numbers it has to be produced. So that so many, if they get, they don't reach the another flower. So at least 1 or 2 will reach. That's the probability, right? If they produce millions and millions of pollen grains, 1 or 2 may reach also is enough for them. So therefore, the pollen grains produced in wind pollinating flowers should be in large numbers, right? Because most of them get wasted. Second point. Apart from that, once they have to get in the air current or wind current, they should be light in weight, right? If they are heavy, they cannot fly in air. Therefore, they should be light in weight is one point. Then they should be dry. They should not be sticky. If pollen grains stick to each other, then they will fall down because they become heavy. That's the reason during the pollination through wind, these plants or these pollen grains are light in weight. They are very light, so they blow like a puff, right? Puff of, like whenever you take something like any powder, if you just blow, then it will go away if it is light in weight. Otherwise, it sticks here only. The same thing, the air, if it has to go, then they have to be light in weight and they should not be sticky, they should be dry, right? This is the important feature. Then, another thing, that is about the stigma. Stigma should catch these pollen grains that are present in air, that are coming through wind current, right? They should be either feathery or they should be sticky. If the stigma is sticky, then whatever comes they will absorb, right? Therefore, stigma will have some other character. They should be sticky or they should be feathery or they should be hairy, right? So therefore they can catch the pollen grains and they can continue for the next further development. Therefore, apart from that, therefore it should be sticky. But apart from that, the stigma should be moving out of the flower character. Suppose this is the flower, imagine stigma is here, and if it is the anther, if this is the petal, the stigma is so much inside that this cannot be attracting inside, this cannot get the pollen grains because it is deep inside. This is a wrong type of stigma character. The stigma should be exposed to air and should be either hairy or feathery like this. They should be hairy or feathery, or it should be sticky, so that other parts and all will be below it, right? Then this can catch the pollen grains which are coming in here, they will catch hold. This cannot do that, therefore this is wrong type. So stigma should be feathery or hairy to help in catching the pollen grains, and the flower should be produced with a longer stalk, right? If the stem is like this and the flower is so small, okay, here is the petal and sepal and other things, leaves and all will be around this, then this flower is not exposed. So if the flower has a long stalk and it comes out of the major other parts of the plant, then it can easily come in contact with air. So this is the important feature for the pollination through the wind pollinating flowers. So what all I told you, flowers should have a longer stalk, stigma should be exposed, anthers should produce large number of pollen grains, then pollen grains must be light in weight, they should be dry and non-sticky. Then apart from that, one more thing you can think, some of the pollen grains will have some wings around them, right? Suppose this is the pollen grain, sometimes they will have some wing-like things here. This will help in flying in air. Like in cycas you have seen, there will be number of winged pollen grains are found in that, right? So therefore wings are very rare, correct? They may be present, helping for pollination, are helping for them, for the pollen grain to fly in air. Sometimes they should have air chambers inside that. Suppose there is an air chamber, they will have more light, they can fly easily. And some modifications are there, they will form pollinia in the calotropis plant, you could have seen. Many pollen grains will combine to form one small sac-like structure. This is called the pollinia. Pollinia will be produced and the insects, it will stick to the leg or mouth parts of the insect. Actually it is a pollination through insects, not the wind pollination. But anyway, this also can fly because it will be having air chambers in that, it can fly in air, right? So therefore, some of the modifications also the plants will do for undergoing the pollination through wind. Okay, so now these are the wind pollinating characters. First you list these, then separately you have to write the characters of wind pollinating flowers, which is 3 or 4 or 5 mark question. Okay, now I will give you explanation about the entomophilous flowers or the pollination through insects. Insect pollinating flowers, how do they modify or how are they looking like, how they appear? Because I told you that plants are those which are silent workers. They don't show any activities, but they are doing everything, right? This is the important thing we have to know, because most of the time we are taking them very negative. They are the plants, we can very happily do anything whatever we wanted, but it's not so. Plants will do every activity and they are far better survivors than other animals, where they will have to search the male and females and all. But here everything happens very silently. Second one I told you, entomophilous flowers. Entomophilous flowers, how do they modify, or what are the characteristics of entomophilous flowers? So as I told you, entomophily is pollination through the wind, the insects, right? So if the insect have to come and get the pollen grains, it should be having some gifts for that, right? That's what I told you. They must 100% have nectar. In wind pollination, nectar is not required. They will not be having nectar. Normally, the wind pollinating flowers are cotton, right? Cotton is a wind pollinating flower. And corn is your maize. They both of them are wind pollinating flowers or anemophilous flowers. They don't have nectar in them. But when you talk about entomophilous flowers, they must have nectar because it is the like giving something for the plant or the insects who are visiting. And sometimes these pollen grains are more nutritious. Why? Because insects will not only simply come there just to collect their nectar, along with some of them should have nectar, some should have pollen grains which are highly nutritious. Right? Therefore, most of the insects will come there, they will live on the pollen grains only, that is their food. Right? And you must be wondering, there are some animal behavior, there is one chapter in which the insects dance they will do. Normally for the mating and all, they will have some dance. Like male insect will go take the pollen grains and make full designs here and all there, looking beautiful and all. Then they will go attract the female. This is how the insects will take these pollen grains and attach their body and they do the makeup. Right? That is also possible. But mostly the insects which they come to visit the flowers, if the flowers produce the pollen grains that are nutritious, they are having some lot of nutrients, vitamins, all that. That's why pollen grains produced in the insect pollinating plants like Amorphophallus and all, they are highly nutritious. And you will be wondering, some nowadays the pollen grain or whatever it is, they will be available in the form of small tablets. Pollen tablets are available. They are available in the small bottles as pollen grains with the nutrition, because all vitamins and all will be there. Therefore, and even so many sports people and all, they will be advised to take this. Right? So keep that in mind. But anyway, let's talk about the entomophilous flower characters. What are the characters of flowers that undergo pollination to the insects? First thing, flowers should be, I told you, it should be colorful. Right? So the flower must be colorful. Flower must have nice color. As I told you, insects are attracted by yellowish, yellow or orange or red color. They are the most attractive colors for the insects. Right? Flowers should be colorful. If the petals are not colorful, other parts get modified like bougainvillea, where the bracts become colorful. Even Poinsettia pulcherrima, the bracts are modified into beautiful, colorful red color structures to attract insects. Then flowers must have nice fragrance or scent which will attract the insects. Most of the time, as already once I discussed, they are not very beautiful smelling flowers, but still insects are attracted, although they are not smelling very beautifully or very pleasant smell, although it's not there, most of the insects like that smell only. So they should have nice fragrance, they should have nectar in them, they should produce nectar. Right? This is the basic thing, the flower character. These all are the flower characters of entomophilous or insect pollinating plants. Apart from this, how insects will come in, and apart from all this, what all it should have is, the pollen grains must be sticky. Remember, pollen grains must be sticky in nature. Sticky means they will stick to the insect mouth parts and its legs and all that. Right? So one of the example is in Salvinia. You just know it is an extra thing about this. Salvinia is a plant, it's from labiate. Labiate, that is almost like our, what is the last name process? Like they will have lip-like petals. So it is labiate family. In that what happens is, and also in some of the orchids. Orchids also it will show like this. Here is the petal, lip-like corolla. And here it will produce one of the anther, one is sterile, one is a fertile anther. Okay? And inside this there will be the stigma. This is the stigma. And this is the gynecium. Okay? Normally whenever insects comes and sits on this, imagine there is an insect which is attaching to this. Okay, this is an insect. When it sits here, then this will turn down and hit the tip body of this. Right? And it will carry pollen grains, it will go away. And whenever it visits the next flower, imagine this is the next flower I am drawing. Okay? This is again going to collect this is the male and female structure. This is a male structure, insect is here. So whenever the insect is visiting this flower, next flower, The stigma which is present here, it will bend down and collect the pollen grains present on the body of the insect, and this insect will be continuously doing this activity. So apart from the anther which is bending down and hitting it, next time when it visits, actually it is not bending, whatever, it's like a seesaw. You know seesaw, like this, whenever you sit here, this will come up, this sit here, it comes up. So whenever insect sits here, this will come back and hits it. It's like a seesaw. Moves here, it comes here and hits it. Okay, like this. Okay. Then whenever it goes and visits the next flower, then the stigma moves down and collect the pollen grains. This is how leviate. This is called as the mechanism of machine, simple machine mechanism, right? Simple machine, like this it is moving. Okay. This is also called as lever mechanism. Lever mechanism is like a simple machine mechanism, right? So anyway, this happens in Salvinia, which is from the family Labiatae. Labiatae refers to, which you don't actually study. It is like having lip like corolla. They come under Labiatae family. Okay. Now, pollen grains should be sticky. I told you already they are sticking. How they are sticking? In most of the insect pollinating flowers, pollen grains are sticky, but they are not very heavy. They are light in weight, but they are sticky. So next is pollen grains should be light in weight because they have to attach to the insect's body parts. Then again, the stigma of the gynoecium should catch hold of this. It should be also sticky or it should be feathery, because whenever insects will go here, suppose this is the stigma, insects entering into this or collecting the nectar is moving down. That time it should collect the pollen grains. So it should, if it is hairy, as the insect moves downwards, that its pollen grains are catched. They are caught here and they are transferred to this. Therefore, they should be hairy or sticky or feathery. Feathery. Okay. Then the endomophilous flowers normally should be exposed to air and they should be beautifully colored. They should have a long, hard, strong stalk. Because stalk, if it is very thin, insect as soon as it sits, it may bend down. Therefore, its stalk should be little stronger, so that insects' weight it can tolerate. Right? So that's about the entomophilous flower characters. Right? Here in this you can give example of Rafflesia arnoldi, which is the largest flowering plant, and it will be producing a kind of rotten meat like smell. Okay? So that's about the entomophilous flowers. Now, apart from this, only hydrophilia I am going to explain you. Hydrophilous flowers, that is, hydrophilous, yeah, just note it down once, right? Once I move out, this can be asked in the neat portions, that is Salvinia, how exactly pollination happens, and other things we have written it here, right? Nectar and all you would have seen, direct example is your honeybees. They go to the flowers just to have nectar, right? Now, let us move on to the hydrophily. Hydrophily refers to pollination through water current, right? Right. So characters of hydrophilous flowers, hydrophilic flower. Okay. So one of the example I'll give you directly by showing you the diagram how it looks like. Right. In plant Vallisneria I'll give you, because Vallisneria plant has it is almost dipped in water, totally underwater plant it is. Imagine this is the ground of water ground, okay, this is the ground. Here the Vallisneria plant is growing. Vallisneria will be having male and female plant separately growing. So this is a male plant I am drawing, this is a male plant, okay. Male plant produce the inflorescence, which is a spikelet inflorescence, like this. The male flowers are produced, right? Spikelet inflorescence. Then this till here you have the water current. Here is the water current. This is totally submerged in water. Imagine this is the male plant. This is a male plant. The female plant grows some other area. Imagine this is the female plant growing. Okay. This female plant will be having spadix inflorescence. Okay. This spadix inflorescence, when it mature, it comes out and it will be waiting for the male to release the pollen grains. This is spadix inflorescence. Spadix means female flowers are here. These are female flowers. Okay. Now, whenever the male flowers are matured, then the male flowers will be released in water. They will come out like this, and they will be floating in water. On the surface of water, they will be floating. So when they come in contact with the female, they bring about pollination. Once pollination happens, this will turn down and it will form the fruit and seeds. So this is how male and female flowers get, they will be separated. Hydrophilous flowers in Vallisneria. Vallisneria I just drawn. Okay. So I hope you understood. In water current also they will make arrangement in such a way that they will reach the other female structure. Actually Vallisneria plant is a dioecious plant. So I hope you know what is dioecious. The male plant and female plant are different. There will be sexual dimorphism. They will be separately located in different places and they will make in some other way to reach each other and bring about pollination. This is what the hydrophilous flower, that is our pollination in water in Vallisneria plant. Okay, so that's a drawing. Just it may be asked for 2 mark also or 1 mark, where you see the hydrophilous character is in Vallisneria. It may happen in Pistil, it may happen in bryophytes also. Bryophytes require water for pollination, you know. But anyway, we are talking about flowering plants, then we will not bring the cryptogams in this. But some of the books are given, it has a bryophytes also, but don't write as much as possible, write angiosperms only. Okay, so this is about the reproduction, flowering plants, that too hydrophilous flowers, how they pollinate. Okay, okay. After this, I will go through the next one. First, you copy the diagram, then let us talk about this later. But you label them. These are all the male flowers. This is the male inflorescence. Inflorescence. Okay, this is the female inflorescence. Okay, because female planting. Okay, draw it and remember it. After finishing the types of pollination and after studying the various characters of the pollination through wind or pollination through insects, pollination through the water current, we will now move on to the next part. We will just go through the NCERT. This is the way they are given. Next part is how exactly these plants avoid self-pollination and help to undergo or always undergo the cross-pollination. Right? Because most of the time you have seen the flowers are bisexual, imagine. Then there is chance that pollen grains are also available, the stigma is also available. But still the plants try to undergo cross-pollination by avoiding self-pollination. So how they do it? Right? Even when you are conducting any experiments, you have to help in cross-pollination. So if you want to do artificial cross-pollination, there are some methods. How you conduct artificial cross-pollination? Why? I already told you the reason for cross-pollination, because cross-pollination brings about variations and new characters and helps in evolution. Okay. So what are the characteristics of artificial cross-pollination? How you do it? Right. First thing, imagine there is one female part. This is a bisexual flower. Here is the male part. This is plant A. Right. There is another plant B. This is stigma. This is the anther. Okay. This is plant B. You have to transfer the pollen grains from this to this, A to B. Okay. How you have to do is, first thing, if you leave them like that, so many times they undergo self-pollination. To avoid this, there should be one thing done, that is removal, cutting and removal of the anthers from this, one of the bisexual flower. We call this as emasculation. Emasculation is a word you have to remember. Okay. After emasculation, you have to take the pollen grains from this and transfer to this. Then whatever pollen grains are produced from here, so after emasculation what you have to do is, you have to cover this with a polythene bag. The flower should be covered, suppose it will be attached to the plant body, covered to the polythene bag, so that this will not have another contamination of other pollen grains. We call it as the pre-bagging or bagging, we call it. Then take one brush, okay, and take the pollen grains from the another one and remove the bag and transfer it to the stigma here. Okay. Then afterwards again put the bagging. It is called the re-bagging or post-bagging. Okay. That's how this seed, when you collect it, it will have the characteristic of B in A. Right. This is the hybrid variety. So the hybrids formed by this, these seeds are called as hybrids. They show hybrid vigor or heterosis. This is what is done by our plant breeders to produce new species, new characters, or to induce new characters in the existing varieties and all. They bring about artificial cross-pollination. This case is very simple. Even you can try at home. If you have any flowers, you have different characters of the same plant. You should not take one hibiscus and put it in the stigma of jasmine. It will not happen because they don't have similar characters. Same genus and species. If you are taking one hibiscus, two hibiscus plants, you have to select one white hibiscus, one red hibiscus. You can try pollinating these by crossing them, right? So don't take the different genus and species. Take the same species. Hibiscus rosa sinensis is the name. All hibiscus belong to that. Take one some pollen grains from one. First you remove the pollen grains from the female structure. Then means that's by emasculation. Take the pollen grains from the other one, put it on this, then bag it. That's done. Then collect the seeds and check. Those seeds, when they grow, then you will have hybridized characters in A and B, both the flowers. Okay. This is a nice easy way to add. Anyway, you people are sitting at home now. You can try this if you have time. If you have plants growing, if you are not growing plants, try to grow now. You can see in front of you they are growing. Just take one old vessel or something broken, put some mud in that and start growing and conduct such experiments. So simple, because John Gregor Mendel did experiments only in his house or in his own church or the monastery garden. He did not go to do any experiment in some laboratory. Right? So all the scientists have started their experiment from home if this is a life science experiment. Right? So therefore, there is nothing like scientists will have to be, this person will become scientist at the beginning is not written. They, by their interest, they become scientists because of their curiosity in nature. Right? So let's now move on to the next part. That is about plants' characters. See, right now I told you how the plants try to undergo cross-pollination, or if you want the new character, you will only help it to undergo cross-pollination artificially. But you will be wondering one important characteristic of plants. What is that? These plants, although they are bisexual, they avoid self-pollination. Always they try to undergo cross-pollination because nature always selects the new variations. Always nature influence cross-pollination. So what are the ways they can avoid self-pollination? That is called as a outbreeding devices. Remember the word outbreeding devices. Means what are the ways outbreeding or hybridization is happening in nature? Means this is also called contrivances for cross-pollination. Another way also they can ask you, what are the contrivances for cross-pollination? Contrivances. Okay? Both are same, right? Then let us learn what are the characteristics in plants to avoid self-pollination and undergo cross-pollination. Okay? That is the outbreeding device to help for cross-pollination and to stop self-pollination. First thing, we learn it as the heading, dicliny. What is dicliny? Dicliny is a condition in which the plants are unisexual. They show unisexuality. So dicliny is nothing but unisexuality. Means the plants always try to be unisexual. Always in evolutionary sequence, if you observe, in plant classification also is done like that. Those who are undergoing cross-pollination, what is that called? unisexuality is always advanced compared to bisexuality. Then what is the unisexuality? So you should know how the sexuality is decided. Suppose the plants have both male and female sex organ in the same flower. We call this as a bisexual flower. Okay, this is bisexual. But this is not dicliny. I am talking about this bisexually. This is a common thing. This is a normal one. Most of the flowers are bisexual. But under this dicliny, I will give you another type. Both male and female sexes are separate, or male and female sex organs are growing separate. It is called as unisexuality. How do they grow? If they grow, imagine this is one plant. Here is the female flower, the stigma, anther or stigma will be there, and here are the another flowers. So this is the one flower. Another flower having only the stigma, anthers are here. So this is the female flower, this is the male flower. If a single plant contains male and female flowers at different parts, different area, that is called as the unisexuality, but as they are found in the same plant, we call it as a monoecious character. Monoecious means both male and female flowers are present separately but in the same plant. Example is your coconut. Coconut you would have seen, one side will be female part, one side will be male, right? So female inside, you would have seen the complete female structures. So this is the inflorescence. Observe. Here are one or two female flowers. All above this you get all male flowers. These are the male flowers. They are present in different positions. This is a female flower, this is a male flower. This is a male flower. Okay. This is called as unisexuality, having single sex organ per flower, but they are growing at different positions. This is called as the unisexuality, but monoecious condition. But what is other dicliny? It is called as unisexuality, but dioecious condition. Unisexuality is true, but I am giving you dioecious character. Dioecious already I told you once in Velisaria. Other than that, you can see in papaya plant. Papaya plant, male plant is different, female plant is different. That's why if there is one male plant in 1 acre of land, it can produce so many pollen grains. All other female plants in 1 acre of land can be pollinated. See, understand how beautiful is nature. And you will be wondering one more thing. Suppose all are female plants, there is no male plant at all, then some of them they can change their sex also. This is how nature will adjust for everything, right? So this is a major thing, you understand this properly, because if all are female, there is no male donor, no pollen grains, then one of the plant which is growing will change its sex, it becomes a male plant. But still, male plant is different, female plants are different. Suppose you have a papaya plant, check it whether it is a male or female, because suppose you have male plant, then you will have only flowers, only pollen grains are released, no fruits are formed. Right? Most of the time they think that, oh, something is wrong with my papaya plant, it's not producing flowers. Why it is not producing flowers is because you might be having a male plant in your house. Right? So therefore, keep observing the nature. The nature will change. Even some animals have the ability, some reptiles will change their sex. If there is no male available nearby, one of the female will become male. That's how always the major aim of every organism is to reproduce. That's why this chapter takes so much of importance in whole of biology. Right? So dioecious character is seen in papaya plant. Even you get it in cycas, but cycas is a gymnosperm, so I am not much going to through the cycas plant. So this is the first character of outbreeding device, that is called dicliny. So they will ask you in the so many questions, what is dicliny? Dicliny is nothing but unisexuality, having single sex organ for a flower, single sex organ per flower. If there is a female plant, female flower, it will have only the gynoecium and the all female parts, ovule will be there. If it is a male flower, it will have only stamens and pollen grains. That's why dicliny is, the unisexuality is having more importance in plant kingdom, because they will always undergo cross pollination. If they have different sex or different area, then this has to pollinate this and no other way for this. Therefore, they will undergo compulsorily cross-pollination. Therefore, unisexuality is the first outbreeding device which can bring about new characters or help in cross-pollination. Right? So, first is diclinic. Let us move on to second thing. Right? First one, remember them. There are 4 or 5 mark questions. Everything, don't just give the meanings and just go on. Everywhere you have to give one example and explain this. I gave you just heading. Along with that, some examples I gave. In that, varieties I gave you. Second thing is dichogamy. Dichogamy is another outbreeding device. The meaning is very nice meaning. If the plant is having bisexual flowers, in them also male sex organ or the anthers mature at different time compared to female. Means both sex organs have a different maturation timing. That's called dichogamy. Di and 2. Right? Gamy is gametes. Both the gametes different time they mature. Therefore, see everywhere the words of the headings are given with some meaning. Don't just by heart them dichogamy, diclinic. I got confused. So many times students will tell me, "Madam, I confused diclinic and dichogamy." There is no chance at all. They mean different. I can't mistake between 2 people who are sitting in front of me. I cannot say you are both are the same. No, I can't. Therefore, every word which is given in biological science has some meaning. Try to go with the words, and if they are Latin words you don't understand, but these are having their meanings in regular common language also. So dichogamy is both male and female sex organs mature at different time. So you have understood that. If suppose in a bisexual flower like this, both are there, na? This only can pollinate this. But whenever the male is mature, the anther is mature. I don't know what is maturity. When the anthers are mature, they will split, their slit will open up and pollen grains are released and the cyst take place. So whenever the female is mature, at that time stigma is non-receptive. What do you mean by non-receptive? First thing is stigma receptivity, I will tell you. Suppose one pollen grain enters of its required time, any pollen grain has entered. If it is of the same type, same species, then the complete, this is the ovules here, they produce one kind of sticky substance by which the whole thing become liquid-like and they allow the pollen tube to develop or penetrate into the stigma. That's called stigma receptivity. So that's a background of this. If the anthers release pollen grains, if they fall on the stigma is normally immature. It is not ready to receive. It is younger compared to the anthers. So anthers mature first, or andration matures first before gynoecium. This is one character, we call it as protandry. What is protandry? It is the maturation of the anthers before gynoecium. Andration matures before gynoecium is called protandry. Pro is first. Andry is andration. So andration matures before gynoecium is called protandry. Right? Sometimes gynoecium will be ready to receive. Right? Gynoecium is ready. It has all liquid and all. That time anthers will be still forming stage. They are very small. They are not yet releasing pollen grains. Then although this is ready, this cannot take its own pollen grain. Some other pollen grains only it will take. Not from this. This condition where gynoecium matures before andration is called as protogyny. Remember, protogyny is a condition which gynoecium matures before andration. I hope you understand. It's very nice. So how they adjust, you just will be wondering. So science is such a subject to at least develop interest, develop curiosity. See how the plants are adjusting. They don't want to undergo self-pollination because it's going to not produce a strong species. What they do? See, so many things are. First they have unisexuality, or although they are bisexual, they are having different maturation time. Androecium available, gynoecium not available, gynoecium available, androecium not available. That's how it is happening. Nature always wants strong species. Even you see in animal kingdom, suppose there is tigers or lions and all there. Whenever they have to mate, before mating, there will be a huge fight between two males will happen. The weaker male either will die or they run away. Only the strong will survive, that will mate and produce strong offsprings. This is what is nature saying, right? Therefore, in plants also always they follow nature's role. The strong species only they want to produce. Therefore, they try to always undergo cross pollination. Okay, so this is second one, dichogamy. Diclining, dichogamy, two things I told you. The third one is hercogamy. That's a third word I am using. Hercogamy is the third one. What is hercogamy? Hercogamy refers to the physical barrier brought between the male and female sex organs, although they mature at the same time. See how wonderful it is. Means whenever the male is maturing at the same time, although female matures in the bisexual flower, physically they go away from each other. How it is? Very interesting thing it is. Right? It is normally observed in some of the plants. Right? What happens is, imagine there is one plant. Its name is, example I will give you, Clerodendron. Okay? In this plant, the stigma is straight, imagine like this, and whenever it is ready to take the pollen grains, then all its anthers will be bending down, right? Whatever I am drawing is an anther. Anthers bend down when the stigma is ready to receive the pollen grains. So what happens? Some other pollen grains only should pollinate this, right? And when the stigma, the anthers are straight, you can see here, the anthers are ready to pollinate, okay? Then very beautiful thing is, this stigma will bend down. See how beautifully they are adjusted. So you don't want its pollen grains, right? Therefore, they will bend the other side. So this is the bent stamen. This is the bent style. This is how they will avoid self-pollination. This is called as hercogamy. So how to define hercogamy? It is the physical barrier that is brought between the male and female sex organs to avoid the self-pollination and to promote cross-pollination. One of the example I give you is Clerodendron. Okay, understood? So now the other one I give you, that is about heterostyly. Heterostyly is another type, right? So the third one I told you, one is dichogamy, first one is diclining, second is dichogamy, third one is hercogamy. Now the fourth one is, it's a nice one, it is called heterostyly. So what is heterostyly? I write it. You will be wondering how it will happen, right? The fourth one I write it now, heterostyly. Means in some plants there are different type of flowers produced with a different size in their styles. Style is of different height. That's called heterostyly. This happens in primrose or primula. Primrose or primula. So how it is? First plant I will show you. The stamen will be, this will be the long anther, or you will write it as a median anther. This is a median anther. This will produce two sides in the styles, two sized stamens. Either it produces the short stamens or it will produce the long stamens. Short ones or the long ones. So this is the first one, what I showed. This is the median style. Okay, let me write here, median style. Okay. Second one, B I am writing. It may be the long one. The style is elongated. Right? This will produce two types of stamens, either short type like this or median type, median height. Okay? This is the second plant B. This is the long style. Okay? Now, third one is the plant with the short style. Okay? This is not medium, it should be very small. Short style, chota wala. Then, this will produce the anthers either median or long. Either long or median. This is C. This is the short style. Now imagine why they are produced like this. So every flower wants to undergo cross pollination so that they will change the style size. That is heterostyly. This plant, the one having long style, will undergo pollination either with the C plant or with the A plant. Right? The one with the median style should undergo pollination with the either B plant or B flower or from C flower. Okay? Then the one C plant or C flower having short style will undergo pollination either with the long style or the B plant or with the A plant. Observe this. So everywhere the pollination is like this, reverse. They will not produce the same size, style and the pollen, the anthers also. So anthers are of one shape, one height, style is of another height. So always they try to undergo cross pollination like this. Understand? So beautiful it is. You have to draw this and just explain two lines or three lines. That's done, right? So every plant will try to undergo cross pollination because of the change in the style length, right? Therefore the name is hetero, means different, styly, having different type of styles, right? So that's a diagram, just you have to draw and explain this. Okay? Now, the last one under this is pollen pistil interaction. In that also they show certain type of outbreeding device. That is the fifth one I will explain you. It is the pollen pistil interaction. What is happening here? Pollen pistil interaction, how it happens. So what happens is normally pollen grains, when they are ready, what they do, and when the style is ready, what it will do, or pistil, what it will do. So imagine there is a female, the bisexual flower. Okay, this you can write anyway. Maybe this is the stigma I am drawing. This is style. Here are the ovules. This is a female structure. Then the male is producing this way. The anthers are here, right? And I told you some of the flowers are closed. Then the whole thing is covered by the petals. We call that as cleistogamy. Cleistogamy, such flowers as in Comelian I told you, they undergo compulsory self-pollination. But I am drawing now any plant, any flower, how their pollen and pistil will interact. Right? And here we have the calyx, corolla. Okay? This is the one. So whenever the female is ready, it is ready to get pollinated, it will secrete some liquid like thing. This will dissolve and get deposited in the tip and ready to get pollinated. At the same time, pollen grains, when they are released, when they are mature, then their stalk or their pollen anther lobe will either dissect like this, or it will open at the tip. There will be small pores present, through which pollen grains come out like a puff, right? Like puff they'll come out. They form a cloud of pollen grains, right? So this is a pollen pistil interaction. So whenever these pollen grains enter here, then they will produce the pollen tube and reach the female gamete. This is pollen pistil interaction happening normally. Normally this is the way they will happen, either self or cross pollen. I told you self pollinating plants, then the whole thing will be covered with a petal. It is called the cleistogamous flowers. But we can see mostly chasmogamous flowers where the sex organs are openly exposed to air. Now, during pollen pistil interaction, there may be a kind of physical or chemical hindrance. That is the pollen pistil, or there will be stop in pollination. How it will happen? Either the stigma will not support its own pollen grains to germinate, or stigmatic surface will secrete some chemicals to kill the pollen grains of its own type, and only when the foreign pollen grains come they invite it. So this is the major problem happening between avoiding cell pollination. During pollen pistil interaction, they avoid by producing either physical incompatibility or they will have chemical incompatibility. So there is a fifth one, what I told you is incompatibility happens. Incompatibility. So what is incompatibility? They are not competing, they are not compatible, they don't like each other, right? Either it may be physical incompatibility where imagine the stigmatic surface is round like this, smooth, okay? If the pollen grain produced, if it contains the outgrowth axils like this, then it cannot stick to this. This will just glide and fall away. This is the physical incompatibility. Their structures are not matching. Second one, it may be a chemical incompatibility. What is it? At the time when its pollen grains enter, the stigma is not ready, right? It may be dry, right? Although pollen grain try to penetrate into this, this is so solid, this will not allow the entry of pollen tube into this. So pollen tube will fail to enter into this. This is the chemical incompatibility. So at the end I explained you about the incompatibility. What is it? It is the non-matching of anthers and the stigma. That is, normally pollen and pistil will interact in this way. When the stigma is ready, that time pollen grains are released, or when the pollen grains of what it wants, that time it is ready to receive them by producing some sticky substance. Normally it happens like this for pollination. But if it is a kind of outbreeding device, or it doesn't want its own cell pollen grains to fall on its stigma, then what it will do? It will show incompatibility. Means it doesn't like its own ones. So what they will do is physically they will show incompatibility. Means either stigmatic structure and pollen grains will not match. If the stigma is very smooth in surface, that time if the pollen grains are highly designed, they will not grow on this particular stigma. It's called a physical incompatibility. Structurally they won't help in pollination. Chemical incompatibility means the style will remain solid although pollen tube begins to form and start penetrating also, style will not open up. Style will be solid and it will not be allowing the pollen tube to penetrate easily. This is called the chemical incompatibility. The chemicals may be released which may not be helping for the pollen tube insertion. Then genetic is, if they are homozygous, then it will help in pollination. If they are heterozygous, pollination will not happen. So this is about the incompatibility, which is another type of outbreeding device. Okay. Now we learned totally 5 types of outbreeding devices. Dicliny, that is about the unisexuality, where male and female sex organs are growing separately. Digogamy, it is the condition in which male and female sex organs mature at different time, so that they will not allow the cell pollen grains to fertilize or pollinate its own stigma. When the anthers are maturing first, it's first one, protandry. If gynecium is maturing before androecium, it is protogyny. Then third one we told about the hercogamy. Hercogamy is physical barrier is brought between male and female sex organs to avoid cell pollination. Then fourth one was heterostyly, where the plants produce 3 different types of flowers having different heights in the style and the stamen, right? The long ones, medium ones and shorter ones. So every time they try to undergo cross pollination. Then last one is self-incompatibility, where cell pollen grains are not allowed to germinate on their own stigma, either physically having certain kind of structural variations or chemically by producing some chemicals which are not allowing the pollen to germination, or genetically they don't allow the pollen grains to germinate on their own stigma. These are the 5 types of outbreeding devices, which is 100% expected question for 5 mark question, or separately 1 mark questions also they are asking in this. So go through them, all of them I separately explained, go through each of them and don't just write their headings and leave it. Practice the examples and drawings and all, then you can have a beautiful explanation also. So once the pollen grains are reach the pollen, the stigma, how it will grow into the stigma? I told you already, that is pollen pistil interaction, right? The pollen tube start developing. Already once I discussed about this when I spoke about the pollen germination, right? This is the exine. From the exine pores, entire will come out to form the pollen tube, right? This you know it. And here the 2 male gametes will pass. These are the 2 male gametes. These start germinating in the stigmatic surface. This is stigma, this is style, this is the ovary. Okay? They start moving into this and later they reach the egg or the ovule. Like this it will be, this is the pollen tube. Pollen tube with the 2 male nuclei, it is entering into the style and they reach the ovule at the end. Okay? This is how pollen tube enters into the stigmatic surface and the style. Right? Now, as they get into it, the pollen tube will be carrying 2 male gametic nuclei or 2 male gametes. Now, once it reaches the ovule, how it will penetrate the ovule? So, next point is the penetration of the pollen tube into the ovule. How it will enter the ovule, right? Imagine this is the ovule. Already I have told you how will be the ovule. Imagine this is the embryo sac. These are the integuments, right? So this is the complete structure of the ovule. It is like this with the stalk. Here is our egg cell. Here are the 2 synergids. Here are the antipodals and here are the polar nuclei. Okay. Now, pollen tube may enter, because this is coming like this, it comes and reaches the ovule. It may enter the ovule through the chalazal end. This entry is called chalazogamy. See, remember the word chalazogamy refers to the entry of pollen tube into the ovule through the chalazal end. Okay. Next is, sometimes it may enter through the sides or through the integuments, either here or through this area. Okay. This entry of pollen tube through the integuments is called as mesogamy. Mesogamy refers to entry of pollen tube through the sides or through the integuments. Then the last one is entry of the pollen tube through the micropyle. You can see here, this is the micropyle. This we call it as porogamy. So there are 3 types of entry of pollen tube into the ovule. One is chalazogamy, where the pollen tube penetrates the ovule through the chalaza. Second one is a mesogamy, where the pollen tube penetrates the ovule through the sides, right, through the integuments. Then it is called as mesogamy. If the pollen tube penetrates the ovule through the micropyle, it is called orogamy. This is a 3-mark question, or separately 1 mark when you ask this. So this is how it enters into the ovule. Now, next thing is how it will penetrate into the embryo sac. This diagram you should know. Then diagram you need not to draw. To understand this, you must know all this, right? Now I'll give you how exactly the pollen tube penetrate the embryo sac during fertilization. Okay, so let me give with that explanation, because that is real fertilization part. Okay, so once it enters into the ovule, whichever way, right? Imagine this is the embryo sac. I am drawing big embryo sac. This is the egg with the nucleus. These are the synergids on either side. Here the antipodals, non-functional actually, they don't do anything. Here are the polar nuclei. These are the things. Okay. Now the next portion is entry of pollen tube into the embryo. This whole thing is a embryo sac. This is the embryo sac. I am not drawing any other things here because membranes and all you already know. Now it has already, pollen tube has already entered the ovule. Now it has to penetrate into the embryo sac. Now it is entering into through this. Always, whether it enters through the chalaza or through the integuments or through the micropyle, to penetrate the embryo sac, the pollen tube has to enter through the micropyle only. Understand this. So, to enter into the embryo sac, it has only one way, that is micropyle. So, it will enter through the micropyle and it will not directly penetrate into the egg, remember, because egg is the future baby, embryo. It can't destroy the egg or it cannot hurt the egg. Therefore, it will penetrate into the embryo sac and open into the synergid. Here it will open up and release the 2 male gametes. See here. These are the 2 male gametes. Now, among the 2 male gametes, one of them will easily penetrate into the egg, fertilizing the egg. Another one will move all the way from here to the center and fertilize or fuse with the polar nuclei. So, once again you have to know, these are the 2 polar nuclei. Polar nuclei are 2 haploid nuclei, N plus N there. Okay. Now, one more male gamete has entered. This is the male gamete. Male gamete is another N. So, this is plus N here. Okay. Now, this is the egg nucleus. This I told you that this is egg. Okay. This is egg. These are the synergids. One of them I drawn here is a synergid only. This is one synergid, this is another synergid. So egg has one haploid nuclei. This will fertilize with one of the male gametic nuclei, male nuclei or male gamete, another male gamete. This is also N in number. So we can understand very clearly 2 things are here. One is, one of the male gamete is fertilizing with the egg, first fertilization. Another male gamete all the way moving to the center and fertilizing with the polar nuclei. So, very important word, that is called as double fertilization. So, in angiosperms, fertilization is said to be double fertilization. Never forget this word, double fertilization. Anytime they can ask you, what is double fertilization? It is the characteristic feature of angiosperms in which fertilization happens twice. What are the 2 fertilizations? First one of the male gamete fertilizes with the egg. Another male gamete fertilizes the polar nuclei. 2 times fertilization is happening. Then, what are these called? Right? First union of the female gamete, the egg with the male nucleus, male gamete, is called as syngamy. Remember? Syngamy, meaning only one is syn is fusion, gamy is gametes. The 2 gametes are fused. It is called syngamy or syngamy. Right? This is the first fertilization. Then the 3 nuclei here, 2 polar nuclei plus 1 male gametic nuclei are fusing. This is called as triple fusion. Why? I hope you understood now. Fusion. That is 3 nuclei, n plus n plus n, right? These are fusing. This plus 1 male gamete, totally 1 n n n. So 3 nuclei are fusing, 2 polar nuclei plus 1 male gametic nuclei together having 3 haploid nuclei fusing. It is called triple fusion. So there are 2 types of fertilization happening. One is syngamy, one is the triple fusion. This double fertilization was first discovered by scientist Navashin, right? His name you never forget because he is the most famous embryologist. Along with another embryologist you should know, whose name is P. Maheshwari, Panchanan Maheshwari. He is the great Indian plant embryologist. Always you remember, and you have to respect the Indian scientists because with this available laboratory facilities they have done so many things. One already I told you about BGL Swami. During microsporogenesis, megasporogenesis he has worked. Then second person was Panchanan Maheshwari, P. Maheshwari. They are all done the great work in embryology. Right. Now he is a great embryologist. He was a great embryologist till today. More of the things, more books we refer from P. Maheshwari. Plant Embryology by P. Maheshwari is a very famous book. Right? Anytime you get a time, chance, just go refer that book. It's a beautiful book, but Indian author. That hardly you get such nicely written books from Indian authors, and he is a scientist also, not just author. Now, so double fertilization is an important word to remember for 5-mark question. What is double fertilization and triple fusion? What you will write? First one is double fertilization is the characteristic feature of angiosperms. You have to write this word angiosperms. It will not happen in any other plants. It is the characteristic feature of angiosperms in which fertilization happens twice. Right? First fertilization is fertilization of one male gamete with the female gamete in egg to produce zygote named as syngamy. I written here syngamy. Right? Second is union of one more male gamete with the 2 polar nuclei forming 3 nuclei together. That is called triple fusion. So double fertilization and triple fusion always very important for examination. With the diagram if you draw, so I am telling you how to impress the person who is correcting is by writing beautiful drawings and understanding the subject thoroughly. Instead of just by adding drawing is like this, 1 round on 3 circles. No. First you should know the subject very well. Then you should draw pictorial representation is always very having intensive meaning in biology. Therefore, try, see all other subjects you are having different techniques to get extract marks, but in biology pictorial representation is very important. So always draw the diagram. That they are not artistic diagrams. You can see here, anybody can draw this diagram, but it has a lot of meaning. That meaning should come in your diagram. Okay, so therefore this double fertilization, if you draw and explain, it has a very good impact. And marks, not only marks, apart from that, your beautification of answers is also important, right? So if at all you have any way, wherever you want to stress, you underline them, or you use some pens, highlighter pens, you highlight them, but don't use red and green. Other things you can use light yellowish and all, you can highlight the answers. This is the way how to score well, right? So this is the total double fertilization and triple fusion. So this is the type of fertilization happening in angiosperms. Now we have to learn what happens after fertilization. What is the end product of syngamy? What is the end product of triple fusion? This is one story, right? First we draw this, points, understand it. Then we will go see in this all the things, double fertilization meaning scientist name, what is syngamy, what is triple fusion, where are the exactly male gametes released, because this is the pollen tube. Pollen tube, although it is entering from any part of the ovule, it is entering into the embryo sac through the micropyle only. Then it releases the pollen grains in not pollen grains, the male gametes and synergids. One of them fusing with this, one of them fusing with the polar nuclei. Together it is forming triple fusion, which is union of 3 haploid nuclei, 3N it is. This is syngamy results in 2N, right? Now, what are the end products? Due to syngamy, what you get is the resultant structure is called zygote. This is a diploid zygote, which is diploid in nature, which in future develop as an embryo. Zygote is the future baby, right? This is going to divide and redivide to form the embryo, and embryo becomes seed, right? This is zygote, very important thing. This is the one which is going to continue the next generation. But what is the fate of triple fusion? Triple fusion is union of 3 haploid nuclei. This is going to produce in future the endosperm. Future it is endosperm it forms. Right? What is endosperm? It is a nourishing tissue for the growing embryo. So end product, this is triploid in nature, this is diploid. So zygote is the future embryo produced at the end of syngamy, and endosperm is produced after triple fusion. Okay, so this diagram you make it pakka, then we will go to the next thing. Okay, so understand, draw and practice. Now, one important thing I would like to give you in this endosperm formation, right? What is that? So keep this in mind. Let me continue with this. About the endosperm. So once the 3 haploid nuclei are fusing here, this is the next diagram. This is a triploid structure. Initially, these all will divide. Free nuclear divisions happen. All triploid nuclei will be freely floating. It is in a watery condition. This triploid condition, we call this as, before becoming endosperm, this whole thing will form primary endosperm nucleus, which is triploid. Later on solidification, it becomes endosperm. So, primary endosperm nucleus is the end product of free nuclear divisions happen after triple fusion. Right? This is primary endosperm nucleus. Now remember one thing. In coconut, you have seen the young one, right? The tender coconut, which is having outer green part, then the fibrous part, then the hard part, right? But when you open up the tender coconut, you get a lot of water. That coconut water is primary endosperm nucleus. Remember this long form, primary endosperm nucleus. PEN, okay? This is PEN. This primary endosperm nucleus is triploid in nature, 3N in number. Then, in that what happens, it has all the nutrients 3 times. There will be growth hormones present, there will be vitamins present, there will be nutrients present, all 3 times. Therefore, coconut drink forms the excellent drink, which has maximum nutrients. You will neither get in any other kind of drinks, either tea, coffee, maltova, this, that, boost, nothing. Only in triple fusion, whatever is formed endosperm, primary endosperm nucleus is highly nutritious. So whenever you want to have some nutrition in immediate way, then have a tender coconut. That is the best and highly sterilized area. See, you can see epicarp is so hard, mesocarp is very fibrous, endocarp, in that it is protected. So therefore it is very beautiful, very excellent drink and very nutritious drink it is. Therefore always remember that primary endosperm nucleus formed in coconut is an excellent drink. That later starts solidifying. So what happens? So imagine this is the triple fusion. All the nucleus develop. The central there will be a vacuole develop, and all these primary endosperm nucleus they start moving towards the end. These are triple fusion nuclei. then slowly what happens at the end, cellularization start forming. There is cellularization forming. Cell wall is brought about at the periphery. This will form your solid endosperm. This is nuclear divisions convert to cellularization. Then this white part, you get the white copra. That is nothing but the solid endosperm. This is not triploid, but this becomes diploid later. This from periphery, the cell wall formation starts. The central part will be still filled with the liquid. So once you have learnt about the fertilization in angiosperms, that is double fertilization, triple fusion, we are now going to talk about how exactly endosperm is produced. Endosperm is the internal nourishing part which is developed from the triple fused nuclei. That is, the centrally two polar nuclei are fused with the male gametic nuclei. Now that endosperm is nourishing angiosperm, right? Now I will tell you the types of endosperms, which is an important neat portion question. It is a neat question. Understand always this, you have to remember, neat question. Why? Because this is in depth, I will explain you, right? So endosperm, I told you first one. Imagine this is in coconut. If you observe in coconut, I told you triploid nuclei will be free floating like this. All the triploid nuclei are floating all over here, right? This triploid nuclei, they are not brought by cell wall formation initially. They will be freely floating in a liquid-like fashion, right? Slowly they start moving towards the periphery, right? Why? Because they develop a central vacuole, right? A big central vacuole develops. All the triploid nuclei go towards the periphery, okay? Like this. Later, once they go to the periphery, the cell wall is brought for them. They become either diploid or they are 2n or 3n. The cell wall is brought from the periphery. So initially there are only free nuclear divisions taking place. Now the cell wall formation starts from the periphery to the center. So this kind of endosperm is called as nuclear endosperm. Nuclear endosperm, where the cellularization is brought from the periphery to the center due to the development of large vacuole at the center. The example for this is coconut. Okay. Second one is there, where this is the complete endosperm area. First divides into two. One go to the chelazal and one go to the micropolar end, and they will be already forming their triploid nuclei. They will get the immediate cell wall. This is cell wall formation starts at the beginning itself. Primary endosperm cells start dividing. First division only converted into the cell, complete cell. Now they further start dividing. Every division is brought about, controlled, or will be followed by the formation of the cell wall. Like this. Both the chelazal and the micropolar end, the divisions will be followed by the cell wall formation. This is called the cellular endosperm. This is seen in the plant like petunia or in datura. Okay. There is a last one. In this, you can see here, this is the first embryo sac. Primary endosperm divides into two nuclear divisions followed by the cell wall formation. So initially it is like a cellular type of endosperm, but later this side, or the chelazal end, they stop division. This acts like a haustoria, and all these triploid nuclei will be freely floating here, and this acts like a haustoria. The haustoria means something that nourishes it. Haustoria, and lower ones will be undergoing divisions. The divisions are followed by the cell wall formation. It is both. It is a nuclear division as well as the cellular division. This is called as helobial endosperm. Helobial endosperm. Okay, this is a rare type of endosperm formed in this. this. Okay, so that's the total story of endosperm. 3 types of endosperms: nuclear endosperm, cellular endosperm, and heliobin endosperm. Okay, that's how endosperm details you have to understand. It is a NEET question, right? Understand thoroughly, and this is the only portion is NEET. Remaining till double fertilization, triple fusion, it is in your regular NCERT syllabus only.